A new study with the Cancer Genome Atlas has identified the key genome of the second most common form of lung cancer. We recently sat down with one of the study's lead researchers to learn more. Dr. Neil Hayes, welcome to North Carolina Now.
Thank you.
You are an associate professor at the UNC Lineberger Comprehensive Cancer Center. You and your colleagues at UNC, as well as researchers across the nation, have been working on studying the genome of the second most deadly type of lung cancer. And you've recently hit a milestone with this project. Tell us about that.
Yes, we have. I'm a member of the Cancer Genome Atlas. This is a large publicly funded project funded by the National Cancer Institute, National Human Genome Research Institute. And approximately 7 years into our project, we finally completed the squamous cell lung cancer, Cancer Genome Atlas project. This was published earlier this month in the journal Nature, and we described essentially the genome of 178 patients with squamous cell lung cancer.
What information does this now give us about this type of lung cancer?
Well, cancer is a disease of DNA. Ultimately, what's wrong with a cancer cell is there's something broken about the DNA. And it turns out that DNA can be broken in a lot of different ways. Before this project, we've had snapshots into the ways in which the cancer genome is broken. But this is the first project that has really taken all of the technologies we have available to us to look at 178 patients at one time with the broadest spectrum of technologies that we could muster to see what's actually wrong with the genomes of squamous cell lung cancer.
Where will this take us in the future? Are there applications for new therapies that could come out of this information?
Absolutely. As I mentioned, this is called the Cancer Genome Atlas, and I think the word atlas is a great analogy for what we're looking at here. In the past, we've had snapshots or essentially sketches of what's wrong in the cancer genome. But now, for the first time, we actually have a map or an atlas where we can really describe all of the alterations of a cancer genome and to start thinking about what are the therapies to direct towards individual patients with squamous cell lung cancer or to groups of patients who might share an alteration. So that we see both individual alterations that could be targeted with drugs, and in other cases we see genes or pathways that are altered in most patients with squamous cell lung cancer.
This is, as you mentioned, part of a larger project. Tell us about the work that you're doing at UNC, because it's not just on this type of lung cancer. You're actually looking at a broad variety of cancers.
That's right. The Cancer Genome Atlas follows on the heels very closely of the human genome, which was published in approximately the year 2000. And one of the reasons to publish the genome, which is the normal DNA that makes us, of which we're all comprised, is to then look at disease states. So one of the easiest examples to study is cancer, because cancer is a disease of DNA. So leadership at the NCI and in NHGRI proposed that we gather large numbers of patients with cancer and describe the alterations in cancer. And when we put that down on paper and wrote the project out, that ended up being 20 cancers, 500 patients from each of those cancer types. And we're now approaching the halfway, more than the halfway mark of our goal of doing 20 cancers with 500 patients with each cancer.
What other types of cancer are you looking at?
So far, we've published the genomes for glioblastoma, which is a brain cancer, colon cancer, ovarian cancer, earlier this month, breast cancer, squamous cell lung cancer, and we'd have to count them up, but we're counting up towards 20, but it includes all of the major tumor types for which tumors could be obtained. Some tumors are a challenge to us. We're doing those too, and that includes tumors like melanoma that are usually small, and there's some technical challenges about which tumors we've taken on.
What is it like for you as a researcher to be studying these cancers and knowing that the information that you're finding can have a direct impact on people's lives?
It's a great time to be a cancer researcher. I can only imagine it's like when the microscope was discovered. that we have a new technology that allows us to look into the disease in a way that we could never do before. Cancer is a disease of DNA. We need to look at DNA, and for the first time we can do this quickly, completely, with high quality. We still have some limitations, but we've never had abilities like we do now. And to look at a disease and get very close to those aspects that will have a direct impact on patients.
Has there been anything in this study that you've found that has really surprised you?
I don't think we have enough time to tell you about all the amazing surprises, but this is an incredible resource that we will be using for decades, I think. But there are a couple of surprises. For example, there's a pathway that was known to be altered in cancer. The pathway relates to two genes, KEAP and NERF. So it wasn't so much a surprise that those genes were altered, but we found that pathway altered in about a third of squamous cell lung cancer. So this is a fundamental observation that if we want to impact squamous cell lung cancer, we've got to understand that pathway. And again, it was known it was abnormal, but we've now highlighted that we have got to attack this or we're not going to understand squamous lung cancer.
And so there's a lot more studying that needs to be done now that we've gotten the genome.
Yes, and sometimes I feel like a painter who's gone into a room and, you know, we've done all the sanding, we've moved the furniture, we've covered the floors, and the room is prepped. And I think we're about to see, you know, the paint going on the wall and really see the changes coming forward. We're not quite there yet. It still looks like a work site, but when you lay a good foundation, I think you can see amazing things coming quickly, and I think that's where we are.
And if our viewers want to follow the work that you're doing throughout this project, how is the best way for them to do that?
Oh, if you put TCGA into your search engine, you will find a wonderful website at the NCI that has all of our updates, and I would start there.
Dr. Neil Hayes, thank you so much for taking time out of your schedule to talk with us.